Right, hello everyone, it's the second video in the sensory ecology series, and today we're going to be looking at hearing and sound in a little bit more detail than we did last time.
No, this is my show, I want to be a star, how dare you! This is mine!
Hang on, Sam, what the hell are you doing? What are you doing this for?
Benito, I'm going to be a movie star!
No,
you cannot take my chance, my dreams!
I'll sort this out. Get up!
Don't you get up! I've told you to get away! Oh.
Perfect. Right, hello everyone! Okay, this is the second video on sensory ecology and the second video on hearing and sound as well, so let's crack on with it, shall we? So, I kind of mentioned this when I was introducing sensory ecology last time, that whenever we're looking at a particular sense, we need to basically ask two questions, and they are how and why, okay? So, how are what we call the proximate questions. Okay, so for example, how does an animal perceive sound? So answers to that may be, having high directionality, high sensitivity, having high frequency selectivity, having a particular amplitude range, frequency range, temporal resolution, all those things that we talked about last time. Okay, so that's how an animal does it. Okay, so that's one question we're trying to answer in sensory ecology. And the second is the why questions, the ultimate questions, okay? So by asking why questions, we're going to be asking why perceiving sound in a particular way, the way that animal is doing it, is evolutionarily adaptive. Okay, so these are the ultimate questions. So things like that could be to find a mate, for example, or to detect a predator, or to find prey. Things like that. And whenever we're looking at hearing, we must also remember that certain animals call in completely different frequency ranges that we do. So we can't just ignore what we can't perceive. Okay? For example, even in the most unexpected places, elephants call at such very, very low frequencies indeed to communicate with conspecifics on the other side of the savannah. Okay? And they've probably chosen these low frequencies because that specific environment is suited to them. They live in an open area, okay, where low frequencies may be favourable. That, and coupled with the fact that larger animals do tend to produce lower frequencies anyway, that's just part of their physiology, there's nothing they can do about that, then that's a pretty good strategy, I should think. And also, as air temperature changes, its sound conduction capacity changes as well. And we're going to be looking at an example of that in just a minute, in terms of bird song. Okay? Now, when sound is propagating and hits an object, there are three things which could possibly happen, and that all depends on the size of the object that the sound is hitting, that wave, okay? So the first thing, if we've got a really, really small object, it could just be transmitted, the sound could just go straight through, transmitted, not really altered in any way, shape or form. By the way, when I say small, I mean relative to the wavelength of that particular sound. Obviously you have really long waves and really short waves. Okay, so that's why sounds with really short frequencies are really good at transmitting through really cluttered environments such as a dense forest. Okay, because they're really short, which means they can get through and get past all those objects much more efficiently than a sound at a high frequency. If an object is really big in relation to the wavelength, then, well, you'll just get reflection happening. Okay, that's pretty obvious. So sound will hit the object, boom, and then reflected at the opposite angle. And it's this sort of technique that's used in bat echolocation, which we will talk about in loads more detail right at the end of the series. Okay, so if an object's not too big or too small, then you'll get what's really important here, and that's diffraction. And for that to happen, the object has to be about 1/10 larger than the wavelength of the sound that's hitting it. Okay, so can't be exactly the same. And it's this which causes directionality in our own ears, as we'll see here, as I've done another lovely diagram. So we've got sound propagating here. The distance between these two lines is the wavelength, okay? That doesn't really matter because I haven't really done this in enough, I've done this very accurately, okay? I'm a very bad scientist, me. But anyway, this is the object, it's hitting it, then you'll get interference happening here now, because that wave has been broken, if you like, and then coming back together again here. But because of that object in the way, and because of the diffraction that's taken place, then you'll get what we call a sound shadow. And that is exactly what creates directionality in our own heads. If you hear something coming from that direction, it's going to hit this ear first and create a sound shadow on the opposite side of your head, right? So that means our brain can work out then that that sound was definitely coming from that direction and not from that one. Okay? Quite simple, really. Another way of putting it is that the side facing the source is receiving an overpressure, and on the other side there's an underpressure of the sound. And you can relate that to all those equations that we were talking about in the last video. Okay, so it's all about this altered propagation of sound within the medium. Another thing that we've got to cope with is that sound produced from the same source, different sound waves may take different routes to reach our ears, so we'll get echoes, if you like. So that creates a problem. So that all relies on the temporal structure of our ears. Because if it's coming from the same source, then the difference in arrival between the different waves to reach your ear won't be that much. So it all depends on your temporal resolution. So this is another thing that our brains have to make sense of. That's called reverberation. And we can use this technical thing, we're not going into the details of it, called Doppler refractometry, okay, to make sense of the behaviour of sound within a medium. Now, one of these key properties that we've got to learn is this idea of atmospheric attenuation. As sound disperses from a sound source, it attenuates in the atmosphere as you increase the distance. Okay? It's an inverse square law because of this thing called spherical spreading. Yeah? As I speak now, the sound waves are propagating in a sphere-like manner. That's how this all works, but in decibels, we can describe this as a doubling of distance being an increase in 6 decibels. But the important point here is that different frequencies of sound attenuate at different levels. So in this diagram here, we've got distance on the x-axis, attenuation on the y-axis. So this one here can't really travel that far at all. If you want to be using this sound, then the things that you want to receive it must be very, very close to you, because it's going to attenuate in the atmosphere very quickly with increased distance. Whereas this fella, maybe this is what the elephants are doing in the African savannah, because look, they've got a really, really low attenuation with distance. So the frequency composition of a particular sound can be changed significantly depending on the composition of the environment. Alright? And this is seen clearly in a study by Roma et al. On katydids. Katydids are little orthopterans, so they're very much related to the grasshoppers and the crickets. Okay? And the frequency composition of their calls was measured at different distances. And they realised that that frequency, if you produce a fast Fourier transformation that we talked about last time, it's very, very different. Because as a katydid in a forest, desperate to attract a mate, you really only want to attract members of your own species, okay? So if there's another katydid out there which sounds a little bit like what you sound like, then that may be a problem. So any strategies to increase species recognition will be favoured by natural selection. Okay? So Of course, the frequency composition isn't the whole story. There's loads of other parts of the song which increase species recognition. Okay, so the repetition rate, for example, maybe the amplitude of the sound, yeah, the rhythm of the sound. Temperature affects the speed of sound propagation, and there's a really neat equation which describes it, and that's this one here. That's C equals 344 plus 0.6 T. Okay, we don't need to worry about how that equation is derived, but just take into account that this is a completely linear equation, right? GCSE math this is. Y equals mx plus c, right? Go on. So C is the speed of sound, T is temperature. And this can be applied to things such as birdsong. Okay, now autumn admittedly isn't the best time to explain this phenomenon because A, the birds don't really want to sing at this time of year, it's just not much point in singing, and B, it's quite cold, so it doesn't really describe what I'm saying. But imagine, we're in the heat of the day, right? It's midday right now, it's 12 PM, the sun's blazing down. At this time of year, at this time of day, it's going to be warmer towards the ground, yeah? Because the ground's absorbing all that heat from the sun, right? And it's gradually going to get cooler as you go up. There's a temperature gradient, right? So that means a bird singing on the ground the sound they produce will propagate faster, right? There's an issue here. Obviously the call can't go downwards, can it? Because otherwise it will be reaching solid ground. It can go forwards, it can be bent upwards. In fact, loads will be bent upwards. But the problem with that is then you get a sound shadow, right? In the middle somewhere along, which means the birds on the ground further along won't be able to hear you as well. So you're not really maximising the distance which you could achieve, even if you are singing on the ground. It'll be much worse if you were singing higher up, right? Okay, so what about during the morning then? Well, in the morning, all that heat on the ground has risen. Remember, heat rises, yeah? So now it's the other way round. It's warmer higher up than it is on the ground. And this is true at night as well for nocturnal animals that sing in the night. So now, a bird singing on top of a tree, for example, can sing, the sound will go fast, reach loads of places, but also it can be deflected up and down, okay? So it can reach loads and loads more birds, and that's exactly what a male bird would want, to increase the chance of getting its mating, yeah? Now, there are other reasons why birds sing in the morning, but this is the one that applies to sensory ecology, right? So, there you go, all explained, and it's that which gives us the beautiful dawn chorus, which obviously we're hearing right now, aren't we? The bloody drill! Thank you.
Oh, for goodness sake, Benito, go away. I'm looking for a mate.
Look at this phenomenon called acoustic competition. That's studied by Roma, Italy in 1992 in bush crickets. Okay, two species of bush cricket were studied, the Hemisaga and Mygalopis genera. Okay, and he found that, well, Hemisaga sings during the day and Mygalopis sings during the night, right? So you might think, well, Hemisaga must be diurnal, Mygalopis must be nocturnal, right? That seems simple enough. Wrong, because a removal experiment was undertaken, so when Mygalopis is removed, Hemisaga sings all the time. That's a bit strange, even during the night. So this suggests to us that Mygalopis is somehow inhibiting Hemisaga from propagating its sound. So it's producing a sound which is maybe interfering with that produced by Hemisaga, making it unprofitable for Hemisaga to sing during the night. That's a bit sneaky, isn't it? So they've reached this sort of balance, if you like, a sort of agreement that, right, you sing during the day, I'm going to sing during the night, because if you sing during the night, you're not going to get any mates, because I'm going to interfere with your sound signal produced. Okay, it's a hard world, isn't it? Another fantastic example is seen in tree-hole frogs. Okay, that's the common name. The Latin name is Metafrinella. So these frogs basically do what it says on the tin. They sit in a hole in the tree and they call to attract females, right? So if you're a male frog, you basically want to produce a call which reaches as far as possible to attract enough females as possible so they can come into your hole. They can come into your hole. Now, these frogs live in the rainforest, and in the rainforest it rains, okay? So the holes of these frogs are going to fill up as it rains, okay? So that means the resonant frequency of the hole is going to differ as the water level rises. What do I mean by resonant frequency? Resonant frequency is when we basically have constructive interference taking place. The echoes that bounce off the sides of the hole will interfere constructively with each other, which doubles up the signal, if you like, making it louder. Okay, so that means you get a louder signal which spreads across the forest. Okay, so as the level of water rises, that resonant frequency is going to change. So if the call of the frog matches the resonant frequency of the walls of the hole in the tree, then you've maximised your signal. You've increased its amplitude, right? And these guys, Lardner and Bin Lakim, looked into this. And they found that the resonant frequency changes with water level, okay, which is what we've already said, and the frogs appeared to adjust their frequency that they called at with the water level. So this was done with loads of artificial experiments. Loads of buckets, if you like, were put up on trees and filled with water, and frogs were put in them, and they measured the frequency of the calls that they produced. So any object has its own, what we call a resonance curve, okay? And what they found was when the frequency of the frog hit the resonance curve, then it would begin to adapt to match it, okay? Brilliant, absolute perfection, aren't they? Amazing. And males were also shown to expend extra energy to produce their call in holes where the acoustic conditions were favourable. Another frog, which we've talked about previously in acquisition, is the Tungara frog. Remember that? The one where it has a brilliant call, it goes chuck! Yeah? Now I think we mentioned in the acquisition series that it was the chuck part of the Tungara frog's call that really gets the females going, right? And some frogs only give one chuck, some other frogs give 2 chucks, and some give 3 chucks, okay? Now, it may make you think, well, if it's the chuck that really gets the females excited, then surely 2 chucks are bigger than 1, right? Or 3 chucks are bigger than 2. Is that true? Well, this was tested, and it showed this effect, which is very common in economics, and it's called the decoy effect, okay? And it's the first example to be shown in any animal. And this decoy effect is all about this choice irrationality. So therefore, when the frogs are given a binary choice of calls, so either 1 chuck or 2 chucks, then naturally they tend to go for the one with 1 chuck. But if you introduce a decoy, a 3rd chuck, right, then they'll go for the one which was originally the suboptimal choice. So the one with 2 chucks. That's pretty cool, isn't it? So the one with 3 chucks, is, if you like, polluting the environment. It's swaying the frog's choice. So now it's going for the one with 2 chucks. Crazy stuff. So that means, yes, sexual selection is happening, but there is a lot more that's going on. The choice that a female makes is not independent of all the other choices that are presented to it. So that means irrational choices are made, but that may be the only option, because rational choices may take too long, so it will lead to lost mating opportunities, maybe even greater exposure to predators as well. So it's thought that this choice irrationality could be leading to stabilising selection in certain populations of these frogs. And this, as a result, will maintain genetic variation, and this is all because, ultimately, because the risk of errors is just far too costly. Okay? So it's not just sexual selection, there's a lot more intricacy to the way the female Tungara frog chooses his partner. Okay, so that's all the behavioural stuff. Now let's move on to some actual ears. How are animals hearing this stuff? These are the two fundamental types of ears you'll find anywhere in the animal kingdom. Okay, so the first one, and I've done some pretty good diagrams here, I've got to say. The first one is the pressure receiver. Okay, and that basically says what it says on the tin. What we have here is a tympanal membrane, like our eardrum. Okay, and we have a sound wave coming in, well, obviously that direction, right? And it's hitting this tympanal membrane. This membrane here is now going to wobble a bit. And you see here it's attached to a sensory cell or a sensory neuron, and that will then transmit impulses. Okay? Brilliant. There's nothing really that difficult about that. That's pressure receiver. And this inside here, see we've got a space here, well that has its own internal pressure. So to set off an action potential, if you like, from this receptor, the pressure coming in from outside must be greater than the pressure on the inside, because otherwise it won't wobble. Okay, so it's all about relative differences in pressure. Now, the thing is about pressure receivers is that they don't show any directionality. That sound wave could be coming from there, from there, from there, from there, from there, and it would still generate the same response by that receptor. Right, here's the second type of ear then. Now you may think that doesn't look like an ear at all, does it? But it is. This is what we call a particle velocity receiver. And now this does have directionality because it's taking into account the velocity of the particles displaced by the propagating sound wave. Okay? Not the velocity of the sound wave itself, but the velocity of the particles. Yeah? What we talked about last time. Now, if you remember back to GCSE physics, pressure is a scalar quantity, just like how, I don't know, speed is, for example. It only has a magnitude, whereas velocity has a magnitude and a direction. So that means it's a vector quantity. So we've got this long, thin hair. That provides a very small surface area for sound to hit it and move it. Okay? And it means that it can be moved from the horizontal side. So it's taking into account the horizontal properties of the velocity of the sound, well, movement of the particles, I should say. So where can we find these particle velocity receivers then? Well, an example is in the arista of fruit flies. Another example is the trichoid sensilla in cockroaches. If you ever looked at a cockroach, you'd see there's a pair of hairs sticking out its arse, right? They are specially designed to detect the acceleration of air produced by the flick of the tongue of a toad, right? Very specific. So once it detects that, you mean it can be looking in completely the wrong direction, then that can generate a fast response and it can run away in the right direction, right? There's no point running towards the toad because then you're dead meat. Okay, so directionality is very, very important if it's a life or death situation. And the problem with these receivers is that they don't really work over long distances. The sound source has to be relatively close to set one of these off. Now the absolute champion, as far as we know, in terms of the amount of receptors you can cram in one spot, is in chironomid flies. Now the Johnston's organ is part of the antenna of most insects, right? And it's good at detecting all sorts of, it's very good at mechanoreception, whether that be sound or just mechanical energy, so touch, right? And its scolopidium, that's the main structure in the Johnston's organ, which detects all these stimuli, has 16,000 sensory cells in them. That's as much as you have in your own cochlea. But look how small that fly is. Incredible. So let's talk about our ears then. We're pretty interested in ourselves, aren't we? Now what's different between our ears and that of an insect is that the main parts of our ear are found right deep into our skull. That makes doing experiments on them and how the ear functions relatively quite difficult compared to an insect, when all the receptors and all the mechanisms behind it are actually really on the surface of the insect, on the cuticle. Okay? Now, I'll be honest with you, I know I'm a brilliant artist, but no way am I going to be able to draw an ear for you. So let's put a diagram up for us, shall we? And let's see if we can explain the basics of what's going on. I mean, we know most of it already, surely. Well, so hopefully we can skip through this a little bit. First thing to mention is that the stapes is the thing which converts that mechanical, those vibrations from the tympanal membrane and transmits it into the fluid-filled cochlea. So now that energy is still kinetic energy, mechanical energy, but now it's in a liquid form. Then on the basilar membrane is where the organ of Corti is, and that's where the sensory cells are placed, okay? They're called hair cells. I can draw a picture of a hair cell. Look, there it is. It's brilliant, isn't it? It consists of one big cilia, if you like, called a kinocilium, and loads of small ones called stereocilia. And here, well, it looks quite similar to the particle velocity receiver, doesn't it? I guess it's kind of similar in some respects. If you tilt it that way, it produces one response. If you tilt the hair the other way, it produces a different response. So quite a similar system in a way, okay? But obviously this is in fluid now, not in air. Now, the interesting piece of information comes in the form of the basilar membrane, which I've also drawn here. I can draw that. Now, what does that look like to you? I don't know, an unevenly planed plank of wood maybe. Well, that's one way to think of it, the basilar membrane in a way, and that's how we get our pitch perception. Okay, no, that's not something you'll find in the World Cup. No, that's a different type of pitch perception. This is perception of sound, frequency. Because as you can see on the left side, we've got slightly thicker basilar membrane than we have on the right side, okay? So the basilar membrane isn't even. So that means if a particular frequency of sound is played, parts of this basilar membrane will vibrate more than others. If a really high frequency is played, then the thicker side will vibrate more. If a lower frequency is played, then the thinner side will vibrate more. Okay? And obviously, as I mentioned, on the basilar membrane is where the receptor cells are. It's where these hair cells are. So different hair cells will be stimulated. So that's how we get our pitch perception, because different hair cells are underneath the basilar membrane, which responds to particular frequencies. Right? Now, an interesting example is this idea of active auditory mechanisms. Now, what do I mean by that? Well, imagine a sound wave, quite a quiet sound wave, so it's got quite a low amplitude, right? It hits the ear. It excites some hair cells. Yeah? Those hair cells are then attached to neurons, which send action potentials to the central nervous system. Right? Now, the central nervous system then sends neurons back down to the ear, into the inner ear, to some specific hair cells. It activates those hair cells, making them vibrate even more. So that will send more signals back to the CNS. So, you see what's doing now? It's being a cochlear amplifier. Of course it is, because it's amplifying the signal. It's making it stronger. So this is particularly useful if a sound source is weak, because the auditory system is actively is using energy, use that to vibrate the hair cells, making the signal stronger back to the CNS. So that makes it easier for us to hear quieter sounds. Ingenious. But however, this will only happen if the signals sent back to those hair cells arrive in phase with the vibrations that are causing the hair cells to vibrate in the first place. So constructive interference needs to happen. They need to be in phase. Right, that's all I've got time for. I'll see you next time where we'll look into a little bit more detail on how animals determine the directionality of sound. How do they know where it's coming from? I'll see you then.